Okay, I think we'll go over like the study guide, and then after that, you can ask some questions, and like, oh, what's on the test? And what's the answer to that? Exactly, okay. Oh, there is one other thing. Hold on. Thank you. Okay. So if you remember, there's 3 domains. This is the highest organizational level of biology. And the 3 domains are bacteria, archaea, and then eukarya. So what's the difference between eukarya and the other 2? Yes, exactly. So if I ask you something on the test about which one has a nucleus, you'll know, right? Okay. Let's see. Okay. Now the cell membrane. This is the membrane that goes around the cell that keeps the outside out and keeps the inside in. And it's made up of what's called a phospholipid bilayer. It's a phosphate with a bunch of fatty acids attached. And it is, let me add something. That's what actually keeps things in and keeps things out in the cell. Now, you don't have to memorize all this, but proteins, of course, do everything. They do all the work, essentially. And so they take care of all the activities in the cell. There's other things, of course, that we've talked about, fats and all that. But the main things that actually do the work are proteins. The thing is, they do all sorts of different things. You don't have to memorize this. But they certainly do all sorts of things like signaling and receiving from one cell to another, so there's chemical communication going on all the time. There's transport proteins like cholesterol, which is a combination of a lipid and a protein together. There is immune system proteins like antibodies. And then the ones that we really are we kind of concentrate here are enzymes which catalyze chemical reactions. Now, a lot of these reactions, the reason they need a catalyst like an enzyme is that the energy required for that reaction to occur is too high. And the cell wouldn't be able to do it. So it uses an enzyme which puts a bunch of little steps in between, and that catalyzes the reaction. And it's everything from cutting up DNA to transcribing DNA, translating DNA, and then, of course, the proteins themselves. So, and enzymes, they have what's called an active site. So there's an area when the protein is folded as an enzyme where the chemical reaction, that's the substrate for what it binds to. You don't really have to know that, but I do. Okay. 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 This is the outside. In. So, this edge right there, that is hydrophilic. It means water-loving. Okay? And then these tails down here, they're actually hydrophobic because they're made of fatty acids. Okay? So they keep water from crossing the membrane. Water can only cross the membrane using certain proteins that are like channels, okay? Or anything else coming in from the outside of the cell, whether it's glucose or whatever it is. So a phospholipid bilayer is, let's go back. Okay. Common enzyme that we're familiar with is lactase. Remember, anything with an ASE is an enzyme. So lactase breaks down the sugar, which is a disaccharide of lactose. When is this gene usually being expressed? Huh? And then in the life cycle of a mammal, when is this usually expressed? Yes, very young, right. So that means mom's providing milk. This is the enzyme that breaks it down into two monosaccharides, glucose and galactose, right? So that's what that enzyme actually does. But the thing is, enzymes, any protein for that matter, they require things to make them and energy to make them and all that kind of stuff. So in general, in fact, for all animals, except for us, the lactase gene stops producing, or the promoter region in the lactase gene shuts off, and the body no longer produces lactase, doesn't need it. Once an animal is weaned, it's never going to come find milk. It might find milk, but it won't be able to really digest it. Of course, with humans, there's some humans that can actually drink it because of the mutation in the promoter region. We'll get to that. Okay. 20 amino acids. There's 13 non-essential. That means our body makes them. And the other 7 are essential, where you have to get them from the diet. And there's some other ones that are kind of quasi-essential. Okay. Oops. Okay. So, obviously, what does fats do? It's a good place to store calories. It's a much better storage you can than, let's say, carbohydrates. Like plants do that. They store most of their energy in carbohydrates. But a lot of animals do it in fat. And you can break down fat, and you'll get more calories out of it. But fatty acids, just remember, just don't do one thing besides store energy. They also are part of the phospholipid bilayer. All these steroidal hormones are all, there's fats in all of them. So there's a whole bunch of things that are fats are used for. Now, fatty acids, they're the same as proteins. Some are essential. That means you have to get them from your diet, and others are non-essential. And cholesterol, as I mentioned, is a lipoprotein. That means lipid, fat, attached to a protein. And it's a steroidal hormone. It actually carries a lipid through well, they function as a unit, and it's able to transport stuff from one place to another. So we use them to build membranes and organelle membranes. So not just the outer membrane, but also the membrane surrounding a nucleus and all the other organelles. All this nerve conduction, all that, hormones, all that. Okay. Now, cholesterol is a big deal. There's an association between cholesterol and heart disease and obesity and diabetes. It's metabolic syndrome, we call it. And it's mainly from our evolutionary history. But in terms of cholesterol, HDL, which is high-density lipoproteins, actually is the good kind of cholesterol. And then the bad kind is low-density, LDL. And then triglycerides are another kind of fatty acid as well. So that's why a lot of people are taking statins nowadays. You've probably heard of them, which keeps your cholesterol in balance. It makes it less likely that that cholesterol is going to attach to the sidewalls of your arteries and then cause a clot later on and kill you.
Are triglycerides?
Yeah?
Are triglycerides bad or good kind of cholesterol?
What kind?
Triglycerides?
Let's see, what do they do? You want to keep them down, I think. But I'm not sure. You can look it up on Google. Okay. So, now, as far as we've gone over proteins, and proteins are basically long strings of amino acids, right? So a polymer. Well, you find carbohydrates in polymers too, long strands of simple carbohydrates strung together like glucose in a long line with hydrogen attached to it. And this is what actually plants make. Plants make carbohydrates. Carbohydrates are fairly easy to digest, so we eat a lot of them. There's simple carbohydrates like sugar, or there's very complex carbohydrates like in potatoes and wheat and the blueberry muffin I ate this morning. Now, a potato is a complex carbohydrate. In other words, long chains of carbons and hydrogens strung together. And the reason it holds so much energy is that when you actually break one of the bonds holding one carbon with another, it releases a lot of energy. Now, there's also stuff like cellulose, which is not digestible by us, but other animals can digest it because of bacteria that they have in their gut. We have bacteria in our gut too, and they do amazing things, but we really can't survive like a cow or a termite on just cellulose. Now, energy, part of the pathway is glucose metabolism, and glucose is the simplest carbohydrate. It's a monosaccharide. Saccharide means sugar. So basically, like potato is a long string of sugars all stuck together. Lactose, milk, is a disaccharide with a glucose molecule and a galactose molecule. So when those are split, we get energy out of that as well. Free sugar, like sucrose, which is a disaccharide, of course, you eat that and your blood sugar immediately goes through the roof. Okay, now, fat, or the energy that we get from glucose, if we don't use it right away, well, it's stored in something that's called glycogen. And it's stored in the muscles. Glycogen is found in the liver and the muscles primarily. So if you remember in terms of exercise, first you kind of burn any free carbs running around in your system. Then it starts getting into the glycogen to burn. And then after that, you don't have anything. You can start burning fat. It's a slower process, though. And then, of course, there's glucose metabolism. And glucose itself doesn't attach to the glucose. Insulin doesn't attach to the glucose molecule itself. What it does is it attaches to a channel that opens up another channel that lets the glucose molecules into the cell. And then the insulin is also in the cell and then directs where the glucose is going. And then vitamins, they're basically coenzymes. That means they usually attach to some enzyme to help it function properly. Vitamins we have to get from the food we eat, except for vitamin D, so we can get, our body makes it if we stand out in the sun. If you're lacking in vitamins, there's all sorts of diseases that go with it. Scurvy is the one that you've probably heard about the most, we talked about. They're actually seeing more cases of scurvy nowadays than they used to, because some people have such bad diets they don't get enough, and so they come in and their teeth are falling out, they're miserable, they have all sorts of problems. And doctors usually don't know what to look for with scurvy, and so they always get misdiagnosed, and then finally it shows up that, oh, they have no vitamin C. And so you always want to make sure that you have a good diet. And not just multivitamins. You can get your vitamins from your diet, and that's really where you should get them. Minerals is the same sort of thing. We have to get them from our diet. They form complexes like hemoglobin has this hemi group, which is an iron molecule surrounded by some other stuff. And that's used to transport oxygen in our bloodstream and then transport CO2 back out so we can get rid of it in the whole aerobic metabolism thing. Vitamins, we use calcium and potassium as ions, where a molecule, an element has a charge. It's either lost an electron, so calcium is, I think, plus, that means it's lost 2 electrons. And then chlorine is minus, and that means it's gained an extra electron. So a lot of these elements are not in their elemental form, they're actually ions. And that's because of electrical charge and getting things to move from one place to another. So if something is positive, and it'll move to an area where there's negative charge. Antioxidants do neutralize free radicals. Aerobic respiration causes free radicals, which are highly reactive elements or compounds. And they can do cellular damage. I'm not sure how much work has been done on it, but they can. The thing is, they actually sell antioxidants nowadays, in pill form. And it turns out those pills don't do anything whatsoever. All the antioxidants you need, you can get from your diet, blueberries, right? A bunch of other things too. Okay, so some questions. What are cell membranes made of? Phospholipid bilayer. What's the function of cell membranes? We've already kind of gone over that. Difference between eukaryotic and bacteria. Does a cell contain the whole genome or just what it needs? The whole genome. So if I ask you on the test something about eye cells, and it'll say, so does all cells have the whole genome, or does the eye only have eye cell genomes, part of the genome? And you would say, well, of course, that's not true. Every cell contains the whole genome. It only expresses part of it. And we went over this in class in terms of, we saw the difference between a muscle cell and a neuron, where some genes are expressed in all cells. They're kind of housekeeping genes for certain processes that all cells must do. And then there's certain proteins that are only expressed in particular types of tissue. Okay. So we'll go more into lactose and lactose intolerance in a bit. So if you wanted to, let's see, saturated fats tend to be straight or kinked, and liquid or solid at room temperature because the carbon atoms are bound to as many oxygens as possible. So, let me. Okay. So saturated fats, that means they're actually straight fats. A whole bunch of fatty molecules all straight together attached to hydrogens. So this is the right one. And then, of course, if we remember, saturated fats are solid at room temperature, and they're bound to hydrogen. So if you want to make an unsaturated fat a saturated fat like margarine, you just add a whole bunch of hydrogen atoms to that fat. Let's see, what was I going to say? Okay, yeah. And so when you add hydrogens, it becomes saturated. It becomes a long polymer again of fatty molecules all stuck together. And the more hydrogens, the more straight it is. And that's why it can actually form something that's solid at room temperature. Also, butter is way better than margarine or whatever they call that stuff, okay? That stuff is garbage. And it just tastes horrible. And butter is just as good for you. Okay. I should do a commercial for butter. So these are all digestible complex carbohydrates like potatoes, carrots, wheat, onions, bread, all that kind of stuff. Pasta. A lot of people, let's say runners, marathon runners, they gulp down tons and tons of complex carbohydrates before the race, and then they've got all that energy that they can slowly use while they're running. Okay. I've already done all this. Fiber, in terms of the diet, fiber is good for one reason. We can't get any energy out of it, right? But it slows the absorption of fats and carbohydrates. So that's why the difference between, let's say, eating an orange versus an orange juice has a lot of sugar in it, naturally. And it goes right into the bloodstream, and your glucose level rises very quickly. But if you eat an orange, it also has a whole lot of fiber in it, and that actually slows the absorption of sugar, the fructose that's found in oranges. Okay, so it's a good way. If you're going to eat, if you like fruit, eat the fruit and not so much the juice. Okay, and then it goes for a lot of even like watermelon is really sweet, and it turns out that if you eat watermelon, it's mostly water, and it really doesn't affect your blood glucose quite as fast. Okay. Okay, so we live in a different food environment than we used to. You might say we live in a different metabolic environment, or different kinds of metabolism work better in certain food environments versus others. Namely, the food environment we live in now is a food-rich environment. You can get as many calories as you want for almost nothing. And you're usually not, also there's a lot of processed sugar, which you would never find in nature, or big fat, fats that you wouldn't really find very much in nature. So people can get all those things now that they weren't able to get in the past. And in the past, if our ancestors ever came across a source that was really full of sugar, let's say like honey, well, they would gobble it all down. They would just, because they usually never get that, and it's a high source of energy, and so what if it's all sugar? They don't care. They want carbs as much as possible. So the environments that we live in now compared to our ancestors are really, really different. And there are alleles that were good in one environment and then are now maladaptive in another. Okay. As far as fats, women usually have more body fat than men because of fertility and reproduction costs. Body mass index. So what's the body mass index? BMI, what is it? Huh?
Ratio of height to weight.
Ratio of height to weight, right. So you want to have your body mass index, I think, below 25. In terms of weight loss, it's the old calories in, calories out model, but that really doesn't explain things. You've probably met people who, well, you go on a diet, the pounds come off, you slowly gain them back, and then after that, your body won't let you lose any weight. It gets very stingy with all the calories. So it lowers your metabolism. It doesn't burn fat. It holds on to it. That's why a lot of people, when they go on these diets, they go up and down, and then they finally get to the point where it's they have to eat almost nothing just to stay the same weight. So it's not exactly calories in, calories out. In terms of, and calories out is how many you burn. Okay, 1 pound of fat is equal to 3,500 calories. Remember, well, it'll be on the test, so you'll use that. Glucose metabolism, ATP. So in glucose metabolism, what you're doing is they're taking glucose and they're turning it into something called ATP. And ATP is the molecule that actually is the energy molecule. So when you, let's say, break down glucose and you do it aerobically, you get a lot of ATPs, about 36 or 38 ATPs. If you do it along the fermentation pathway, let's say you work out a lot and you have lactase acid buildup, you only get 2 ATPs. So in aerobic respiration, we get a lot more ATPs from our food, and so it can actually fuel, like if we didn't have this aerobic respiration, we would not look like we do. And we would not have high body temperature and all that, because 2 ATPs from each molecule of glucose would not be enough to sustain us or sustain cellular function. So there's something called, so there's aerobic metabolism of glucose and anaerobic, which means without oxygen. With heavy exercise, there's usually a point where you've burned up all the available carbs and glycogen that you have, and you hit a wall. And that's kind of a threshold. And some people can power through it, but basically they're not doing aerobic respiration anymore. 3 things that are required for muscle building. So, you can't do muscle building and losing weight at the same time. Let's see. So if you took an obese person with diabetes from today and put him or her with a group of humans from 30,000 years ago who are all experiencing famine, would she be more likely to survive than a skinny person from today? And the answer is yes. This whole idea of glucose, I mean calories in versus calories out, is not true. Some people today have gotten alleles from their ancestors, and these alleles protected them from starving to death. They made their metabolism very stingy. People like that in the past, when there was a food shortage, they were the ones that would survive. And all the skinny people would die. Nowadays, it's different. So nowadays, we live in a completely different food environment. There aren't famines, for the most part, at least not where we live. I mean, they might run out of your favorite ice cream, and that's kind of a famine, but not to the point where you're going to die. So nowadays, that kind of stingy metabolism is actually selected against instead of selected for. And that's how evolution works. Evolution, that's why there's no trait that is perfect in all environments. It might be good in one and bad in another, and this is a perfect example of that. Okay. Okay, so if you're going to figure out, let's see, a male bodybuilder with 10%, I know what we did, okay. Okay, so it's question 1. Who would have the higher BMI? A male bodybuilder with 10% body fat, or a man of similar height with average muscle mass and 12% body fat? Yeah. Muscle weighs more than fat. So that's why BMI is not really such a good measure, because it gets confused with muscle mass. So you can have a bodybuilder, let's say Arnold Schwarzenegger, his body mass index would have been gigantic, and yet he didn't have an ounce of fat on him. He just had muscles on muscles on muscles. If you wanted to lose 2 pounds in 1 week through diet alone, how many calories per day would you have to cut from your normal dietary intake? Well, 1 pound in a week, you need to lose 3,500 calories, which would be 7 days, 500 calories per day. If you wanted to lose 2 pounds, it would be 7,000 calories, so you'd have to lose 1,000 calories a day. In other words, cut 1,000 calories out of your diet. That's why diets are so terrible. What makes most ATP? Aerobic metabolism. You're on a hunger strike. You test increasing fees in the CSU system. Where do you think your energy is coming from on day 1 of the fast? Glycogen and fat, or only, so it's glycogen and fat. Day 5, only the fat is left to burn, and then day 10, we start getting into anaerobic respiration and fermentation pathways. And so you actually start breaking down protein. You can diet to lose fat and exercise at the same time, and that's false, you know. Okay. So, in terms of genes, we know a little bit about DNA. We know that there are these bases that we talked about, 4 of them, AT and CG. These are the bases that make up a gene. And each 3-base pair codes for 1 amino acid of a protein. So, as remember, genes don't code for traits. They code for proteins that then make up the traits that we have. Like blue eyes or brown eyes, it's this pigment, it's a protein pigment. Okay. There's also, it turns out only about 2% is actually coding for proteins of our whole genome. The rest of it, well, it used to be thought of as junk. Old genes that have been turned off and are just hanging out, and some other things. But it turns out now that a lot of the genome is actually part of the regulatory process of genes and gene expression. Of course, genomes are related by inheritance, so you're much more likely to share the same genes from identical by descent from getting them from your parents than anywhere else. Okay. Remember what the structure is. Sugar phosphate backbone, you don't have to know about that, but you do have to know about the bases. And so if I gave you a string of, we'll get to that actually. So a codon is 3 nucleotide bases for 1 amino acid. The chromosomes are, of course, contain the genes and the regulatory regions and all that. We have 23 pairs of chromosomes, 1 copy from our mom, 1 copy from our dad. They're all identical except for the sex chromosomes. Most of the DNA, it turns out, is in the nucleus, but I will say that mitochondria actually have some of their own DNA. And that's because mitochondrial, at one time, were a kind of bacteria that then became part of a eukaryotic cell. And then short tandem repeats, you guys remember what those are. You have a long stretch of repeats, let's say TAG, 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 TAG strung together. And in a certain location, one person might have 8 of those TAGs all strung together, another might only have 2. So in that one location, you can differentiate between one person and another. And then if you use like 15 or more locations like that, the likelihood of actually having the same STR pattern in all those locations is infinitesimally small. It'll never happen. I think I showed a 1 in a billion. It's actually much worse now in terms of the chances of that happening. So that's what they use, and they get the DNA from our tissues. You can get it anywhere. Hair sample at the very base, just doing a mouth scrub, whatever. And then you can take that DNA and you could do something called, what is it called? PCR, right? Polymerase chain reaction that will amplify it into millions and millions of copies, and then you can do the analysis, like the STR analysis on it, with a gel electrophoresis. They used to only use 13. They do more now. Actually, now they use something called SNPs, which are single nucleotide polymorphisms, but you don't have to know that. And then, of course, you use these short tandem repeats in a gel electrophoresis, and so a person might have 8 at one locus and another person might have 2. Well, in the gel, the longer one is heavier. It's going to move more slowly down the gel than the shorter one. So you can separate into bands. The 8 one will be up near the top, and the 2 repeats down near the bottom. Okay? So they'll move at different speeds. And STRs, or DNA, is actually negative, so it'll move from negative to positive. In terms of a person will always have 2 bands. One from mom and one from pop. And so you have to know how to figure out who's, let's say, the mom and dad, or who's the baby, or whatever, by the bands that they share, the SCRs that they share. Okay. So for the DNA sequence AA, GCTCT, you want the complement to it. So remember, in DNA, there's always going to be the base pair and the complement to that, A and T, C and G. Okay? So you'd end up with TT, CG, AGA. Okay. So we can determine if a DNA profile. We can get DNA from all sorts of places. Let's say you have a DNA suspect, does not match the DNA profile from cells recovered at the crime scene on the cigarette butt. What can we conclude if no DNA recovered from the crime scene matches the DNA profile of the suspect? So what can we conclude? Anything?
If the DNA does not match, that means the suspect is not the suspect.
Well, okay. So, but we can only say that about the cigarette, the stuff on the cigarette. Okay? Yeah.
Probably say I got a sibling.
Huh?
Probably say you have a sibling, like a twin,
A sibling?
like a brother.
Oh, yeah, you could have something like that. Right. So, let's say, actually, if you had an identical twin, and you weren't there, but your identical twin was, and they found the DNA, then your identical twin, who's the evil twin, would then send you to jail, saying he was innocent, you were guilty. Not you. Is your identical twin evil or nice?
This is evil.
Okay, that's what I thought. Yes, I knew it. See? Okay. So, really, all we can say is, let's say on the cigarette butt, there was no DNA from the suspect, so the suspect probably did not smoke the cigarette. Now, you could actually smoke a cigarette while it's not getting any DNA on it, but let's forget that. The suspect still, of course, could have been at the crime scene. You have to search all over the place for DNA. So he might have been at the crime scene, you just don't know it. By just saying, oh, I've got a cigarette butt and his DNA is not here, so he's obviously not the criminal. You just said, well, at least we know. Also, let's say you find out that the suspect hates cigarette smoke. Well, of course he wouldn't have a cigarette butt, right? But he still might be there. Okay. Okay. So, given the STRs here, what are the possible banding patterns of the child? Okay. We'll do mom first. And then we'll do dad. So we've got a dad up here. Oops, sorry. Okay. So we've got the dad up here. And we've also got, let's see, the dad down here, and the mom there as well. Okay, and then we've got mom, and then dad, or dad and mom. Okay? Now, oh, by the way, sometimes you'll have a really thick band at the same place. That means they both have the same allele, the mom and the dad. Exactly like right here. So, if you actually had a band here, it would probably be extra. Good. So you can figure out what the next thing is. Okay. And then, so gene expression. So as you remember, you learned that genes code for proteins. But there's also a large part of the genome that actually causes or regulates gene expression. How much gene product is made, when it's expressed, where it's expressed, all that kind of stuff. And so, first you have to know exactly the process of going from, let's say, DNA into a protein. And as you remember, you go from DNA and then you transcribe it into RNA, and that RNA is then translated into an actual protein. So the DNA as a molecule does not do anything. It just provides instructions. it is the proteins that do stuff. So there's a bunch of proteins involved when you're copying DNA into RNA. It's called RNA polymerase. And it adds all these nucleotide bases in the correct order. And this is the messenger RNA that then is ferried out of the cell into the cytoplasm, and then it becomes larger, this large complex cover called a ribosome, which then takes that mRNA and the instructions on it and translate it into the amino acids that make up the protein. Now, in terms of gene expression, the protein, the gene that codes for the actual protein, won't do anything unless it's told to do stuff. And this is where gene regulation comes in. So most genes have what are called promoter regions and enhancer regions. Promoters are usually just upstream, and they are like, not exactly like switches, but like dimmers. So let's say you have a promoter for lactase. It switches on and it goes up to a certain amount of the lactase protein that it wants to make. And that's in the promoter region. Now, there's also other regulatory elements called enhancers that are far away in sometimes on other chromosomes. But for the most part, for us, we're talking about the promoter region, which is usually right next to the actual gene. So that's why with mammals, once they're weaned, that promoter region that's keeping the expression of lactase, it's been turned off. You don't make lactase anymore. Okay? So if you found, okay, you have someone who's lactose tolerant. That means their body is making lactase. So where would the mutation be for that to happen? It wouldn't happen in the structural gene. It would happen in the promoter region. In this case, someone had a mutation that kept the lactase gene producing lactase. Now, for the most part, that would be bad because once mammals are weaned, they don't need lactase. It's expensive to make lactase. If you're making lactase and you don't need it, you're at a disadvantage. But in the correct environment, namely where there's dairying and cows and things like that, a person who has that mutation suddenly has a food source that everybody else doesn't have. Oh, in terms of the nucleotide bases, for DNA it's different than it is from RNA. So it's identical except for the T in DNA is replaced with a U on the RNA, messenger RNA. So you have to remember that, but that's about it. Otherwise, a U, inserted in for a T, it codes for a certain protein or a certain amino acid. Okay. So this goes into all the differential gene expression. And then the whole lactase persistence thing. Anybody watched that video? Okay, some of you. The ones that didn't, you're out of luck. Okay, let's see here. Okay, so GMOs, or genetically modified organisms, That means you've actually taken the gene from one organism, put it into another, to get that other organism to actually express that protein. So if we put in the human insulin gene into a bacteria, which is the transgenic organism, the bacteria or the human? Bacteria. If we want, let's say, goats to make spider silk, we insert the spider silk gene with a goat promoter in milk duct cells, and then it makes the protein, the goats actually make the protein in their milk. And then you can get all that spider silk, which is very useful for a lot of things. It's got amazing properties that things like steel, it's stronger than steel, but much more flexible. So GMOs are basically genetically modified organisms where we've kind of put in new genes to get them to do new things. There's a lot of controversy about that. I mean, in some cases, it's obviously a good thing. Let's say you had a lot of people have difficulty getting vitamin A in their diet. And one way to do that, though, is you can take a gene that actually produces, let's see, what does it do? Oh, it produces, what is that substance called? It's an anthocyanin that are made by plants. And you can put that gene in the rice, and then you'll get rice that actually has the precursor to making vitamin A. So that's pretty cool. You can also do a lot of things, let's say with, let's say corn. You want to make it resistant to certain insects and viruses and bacteria. You could do that. You could also make it resistant to an herbicide, so you could spray a field with herbicide, kill all the weeds, then plant corn. Now, the corn itself is just like any other corn. Mostly it's not like the corn you or I eat, it's the corn that we feed the cattle. But it's got the same nutrients and all that. The only thing that's problematic, perhaps, is that it's got all these new genes, and sometimes plants hybridize really easily compared to animals. And so you could have these genes that actually escape into a new species. So there's some questions about that. I think in some cases, it's a good thing. In other cases, you have to be careful. And we do it with what's called recombinant DNA technology, where you actually use enzymes to cut a certain place on the genome to then insert a new piece of DNA. Except that now we're using a new system called CRISPR-Cas. And CRISPR-Cas lets you precisely cut along a DNA sequence or an RNA sequence. So with recombinant DNA technology in the past, it was hit or miss. Sometimes you would have to do it, a million times to get a few places where it was actually inserted into the right genome. Okay. Oh, we should start. Okay, 1 second. So just remember about CRISPR-Cas. It's a very, very efficient, precise gene editing technique. That's it, I think. Okay, you guys have any questions? Yeah. What's the what? Oh, the gene gun. Okay, so you can actually shoot genetic material into a cell or plant cells, let's say, and do it that way. It's not being used anymore with CRISPR, now that we have CRISPR-Cas. Any other questions? Yeah.
Are we gonna need to know what the A, T, C, G, and U stand for? In the
Oh, you mean the actual,
word?
yeah, no, just make sure you know the complementary base pair. Okay? Okay, okay. Let's see, I think there's other people coming here, but we'll start. Okay. Why don't you take that half, and I'll take this half, and we'll see where we end up. Okay?
Do you want me to hand out one by one or just by rows?
By rows. The main thing is that they're in order, so that people aren't, all doing all that kind of stuff. Yeah.
Do you want me to take the test with this class, or what do you want me to do?
I'll give you that stuff here right now. Okay.
This is over there.
And then, let's see here. Okay.
Not a whiteboard.
Oh, yeah. It doesn't matter. They're all the same in the back. Okay? So, just answer the ones you didn't. Okay. Oh, good. Give her some of yours. Okay. And just pick rows, and I'll start over here. Yeah?
Question, you have it right here, how many amino acids can be coded in DNA, and it's supposed to be 12, right? But it's like,
Oh. No, so how many is 4? Because there's a codon is 3 base pairs long, and it's 12.
oh, got it. So that's what she's telling 12, like,
Yeah.
There's one more paper behind it.
Yeah, we can start. So make sure on the Scantron that you put the test and the letter. So it's test 2A or 2B or 2C. Okay, yeah.
are we able to write on the board?
Yes. So you can write on the test as much as you want. Okay. Who doesn't have one? Let's see. Oh, okay, let me get. Anybody else? I have a test. Okay. Okay, so, yeah,
Can we write on the exam?
yep, I said, so you can write on the exam. You can stick a corner of the exam in your mouth and chew on it. I don't care what you do. Okay? Oh, put your name on the exam too. Oh, at the very back, there is a figure. There are actually two figures of the gel electrophoresis, right? Yeah. Oh, yeah, you shouldn't do that. Here, let's give you something different. Give me a yellow one.
The white one.
Oh, on a white one, never mind. Okay, a white one. I knew you were trying to get away with it. Okay. Okay. So, on the very back page, there's a figure of two gels. They correspond to two questions in each test. And on the back page, it says which questions. So depending on which test you have, it'll be that number question. Okay? And then there's also 10 extra credit questions. Do all 10, but you can get up to 6 extra credit points. Actually, 12 extra credit points. Okay? So you're getting extra credit. I would answer, you don't have to answer all of them. You could just answer 6, but if you miss one, then you miss one. Whereas 10 chances, 6 out of 10 is better than trying to get 6 out of 6, right? So do the extra credit. If you have any questions about something you don't understand, like, oh, what's the answer? I'll just come on up. I'll talk to you. Okay. So good luck.